Welcome to the Beyond the Reef podcast, where I talk to experts and researchers in the reef aquarium hobby, discussing a broad range of topics from corals and reef biology to water chemistry and equipment. We take a deep dive into our guests' methods, techniques, and top reef keeping tips. My name is Adam Sutherland, and I am the owner-operator of Frag Garage Corals, based out of British Columbia, Canada. Hey, what's up, everyone? Welcome to today's episode. I am joined by two PhDs, also husband and wife couple, Dr. Mareen Möller and Dr. Samuel Nietzer, who do work on coral reproduction and spawning out of the Oldenburg University in Germany. A lot of this research builds around the Jamie Craig's Coral Spawning Lab research, so if you haven't seen my episode with Jamie, I would say go back and watch that one and get well versed in all of the stages of development before we get into this one. With Sam and Mareen, I really wanted to get into a lot of the very specific areas they've been working in this space. And one of the cool things is that they have developed these hybridized captive raised corals that they plan on bringing to the German market for sale. So you can check out their website. It's called SciReef. I'll leave a link to that in the description of this episode. This podcast is now officially brought to you by Fauna Marine. I use a lot of Fauna Marine products myself, as well as their ICPs, and I think they are an excellent company, and I definitely get behind what they do. I wouldn't take on a sponsor if I didn't. Also, if you're looking for ICP consulting through Fauna Marine, I will be an ICP consultant soon, so you can select me from the list and book a consultation with me. As per usual, I ask that everyone hit that subscribe button, like, share, and comment on this episode. Let us know what you think. And if you want to send me a tip, make a financial contribution, I have a Patreon, which is patreon.com/beyondthereefpodcast. Or you can also send a PayPal at beyondthereefpod@gmail.com. I hope you enjoyed this episode, and please let us know what you think. I guess a good place to start is, are you guys hobbyists initially that kind of got into this? Like, can you kind of give me like an early stages how you got into this?
Yes and no. So that's basically the short answer.
Yeah.
So I've been a hobbyist or aquarist for pretty much almost as far as I can think back. So I got my first tank I got when I started elementary school, freshwater tank with the guppies and the usual stuff.
Pretty standard.
I always had that fascination for aquatic critters. So I had always aquaria. I got my first seawater tank at 16. Back then was all metal halides and a lot of makeshift stuff, a bunch of soft corals, so kind of primitive, but always stuck to the hobby. And then when I got into university, I had also a coral tank at my student apartment. And but by the time we got more into the coral science part and there was more traveling involved, I had to give up my own personal tank. But soon after, when we started to work here at the University of Oldenburg, there was the new aquarium facility being set up, and in 2012, 2013, I took over the management of the facility, which I'm still running to this day. And then I was back into the marine aquarium fun.
Yeah,
my hands in the water.
Cool. So you kind of started as a hobbyist, got super busy with school, and then you were able to actually be a hobbyist kind of as a profession in a way too, at a high level. So, and Mareen.
Yeah, and I was never really a hobbyist. I always wanted to be a marine biologist. I always was interested in the science part. And that came way later. Once we got together, we met at university, we became a couple, and I took care of his aquarium when he was on long holidays. Then it started as like really the interest from the scientific side. And it's necessary to keep the corals happy. And then we were really lucky that Sam already had that background, because in the university setting, there's basically no people that actually know how to keep a coral. Very, very few that actually know how to keep corals happy. So without Sam's background, and obviously would have not been able to do the scientific work we were able to do.
Yeah, it's good to have the blue thumb for sure, as they would put it.
Yes.
So you were interested. You were doing marine biology, but you didn't really necessarily know it would go in this direction as far as farming and spawning corals and stuff like that, I imagine.
Oh no, we got the idea way later. So we started working with juvenile corals, with coral larvae, with coral settlement. We were really interested in how like all the stressors we put on corals. We know quite a bit what we do with adults, but we know very little what we do to how the juvenile stages react. So that was really a lot of our scientific focus. So to do that, you have to generate juvenile corals, which is not that easy.
No,
but
clearly.
we became better and better at it. And the 1 year we had like 30,000 juvenile corals, juvenile acroporids, and Sam, being from the aquarium hobby, he said, like, Mareen, do you have any idea what that would be worth on the market? And I was like, no clue. I guess so. So, that kind of was how the idea was born. We actually, the methods we started to develop could become
and just because we haven't mentioned that the methods, the coral spawning and all that didn't happen in an aquarium setting, but that happened in a field station. So we started to work with Professor Peter Schupp, who used to be a professor at the University of Guam. That's where we met him first, the island chain of the Marianas. Basically, they have a really nice university, marine biology institute. So we used to go there almost every year since 2010. For the mass spawning in summer. So that's where we started to work with the coral spawning.
Okay. That makes sense because I saw in your Skype thing, it said Guam, USA. And I was like, that's not right. You guys are Germany. But you must have spent a fair bit of time there. And I bet that was a pretty cool place to do research and probably like live for a little while, hey?
Oh, it's an amazing place for sure. It's definitely our second home. We've been there on and off all the time. Way less now in recent years, but I was lucky enough to go last year for a couple of weeks. But it used to be a much longer trips always for the summer spawning, and we used to go there almost every year. So in 2011, our professor Peter Schupp went to switch to University of Oldenburg. So we stayed in his lab and basically have been ever since. We did our masters in his lab, then PhD, postdoc, and so on. So we used to travel back and forth to Guam. To continue the work with the juvenile corals, settlement cues, and effects of temperature stress and all that. But then, I don't know if you probably remember, but there was a pandemic hitting.
I seem to recall.
2024 something, right?
Yeah.
That had a pretty bad impact on our travel plans. Basically. So that was all,
I would imagine that period of time probably gave you a little bit of time to focus on your research at home too, though. I mean, you probably made quite a bit of progress here.
Without the pandemic, we wouldn't have made as fast or as progress as quickly as we did with like XC2 spawning because at the time we had a collaboration, or we still collaborate with the Tropic Marin. We had a research project between the University of Oldenburg and them that aimed to optimize methods of spawning and rearing. And at the time when the traveling was out of question, luckily Jamie Craig started his company producing spawning systems. And so we had him and Vince and Mike build systems into a remodeled or an old shipping container, basically the lab in the box.
And the instructions, the instruction manual.
Exactly. With the financial support of Tropic Marin, we were able to obtain the container. And then actually start the spawning operations here in Wilhelmshaven.
Awesome. Yeah. So, I mean, obviously, Jamie Craigs is sort of an early innovator of a lot of this stuff. And
absolutely. Like, we wouldn't have tried without the knowledge that he was able to do it. Because it's very, very different. Like we were really good doing these things in the field, but of course it is different doing it in an aquarium setting. Very, very different. So he was really the first one to do it, and all the others are just following.
He's the OG coral daddy.
Yeah, for sure. I guess some of the things that like I've detailed, I mean, back to the episode I did with Jamie, it's about 2 hours and 40 minutes long. It's long, but we go through every, part from the settlement, the spawning to the settlement, right through, all the different species he's worked with. So I think we probably won't discuss some of those details, but what I'm going to want to try to get into with you guys is some things that you've maybe discovered on your own or done differently Because it seems like you've produced some hybrids that look pretty damn cool, and like that might be where, you know, some of your energy is focused on. I bet you guys talk to Jamie a lot as far as exchanging information. You know,
yeah. We met him the first time actually on Guam when he was there for a workshop in 2014 or 13. I think 14
or 15, I would say,
okay.
but yeah. A
One
little while back for sure.
Yeah.
So, we got along really well right away. He's a great guy, and he's just so passionate for the coral work, and it's so great what he's done for sure. I mean, his focus is really strongly on like figuring out how it works, how the system works, how they are spawning, how you can manipulate it, and he's done amazing work in that regard. So that is not really our focus. So we really want to try to get productions up for aquarium purposes, but then also later on maybe for reef restoration purposes, because it is one thing to get the corals to spawn. It is not easy, but I would say a lot of people that can keep acroporas really happy would be able to do it when they set up a system like that. But this is really just the beginning. Having the coral spawn and then having the embryos develop is a little bit tricky already. But then having the larvae settle in high numbers and then having the recruits develop in high numbers, survive in high numbers, this is the trickiest part, I would say.
Yeah, so that actually brings me to a question. There was an article I found on the Oldenburg website, and it mentioned that you guys were working on a specific material that was more, I guess, ideal for settlement. Can you kind of tell me a little bit about that and kind of how you came to that?
Yeah. So the settlement process is, we have been studying that, or the group of our professor has been studying that for around 15 years or even longer. I mean, that settlement is one of the most crucial points in the life of a coral, right? The place where the larvae attaches and metamorphoses into a polyp is where it's going to spend the rest of its life, right? So if this spot is a good spot, the life can be potentially hundreds of years, or at least tens of years. It's a bad spot, it can be over in a couple of hours. So they have to make a really distinctive, very good decision. And this decision is mediated by chemicals. I'm not saying it's exclusively the chemicals, but chemicals play an important role in that. And these interactions is also the interactions between chemical compounds and biology is one of the main focuses of our working group. It's called the environmental biochemistry. So that's basically right in the focus of the research. So as Jamie also said in the episode, the crustose coralline algae are a preferred settlement substrate for the coral larvae in nature and also in the lab, of course.
Yeah.
So on these CCA, there is a specific microbial community, right? So and over the years, we've been isolating bacterial strains, isolating them like in individual strains, more than 100 over time, and tested them individually, whether or not they would induce the settlement of coral larvae. That was actually a Mareen's bachelor thesis that she did on Guam, isolating the CCA bacteria and then testing them individually. There was one specific strain of the genus Pseudo-Ultramonas. It's like a pink growing bacterium that induces the settlement very reliably or very effectively. And over the years, we were, of course, not just the two of us. There's a whole team of chemists and people involved and other PhD students. So it was a big operation to fractionate or to extract all the chemicals that the bacterium produces, and then go step by step and try to figure out which molecule is involved or is inducing the settlement. And then by 2021, the molecule or a molecule could be isolated and identified that induces the settlement in a whole variety of species. So not just the acroporids, but also the Leptastria and other brooders. So it seems to be kind of a universally potent settlement.
Okay, so just as a little make sure I'm following. So there is a molecule that has to do with when the larvae is comfortable settling, essentially, right? But you also said that there's a bacterial strain it's competitive. Did you say it was competitive, or was it what was the relationship exactly?
No, so there is the CCA. On top of the CCA, there's a bunch of different bacteria, And some of the bacteria, or at least one of the bacteria, produces one compound that we isolated from him that induces the coral settlement.
Induces the settlement.
So it might be more than that one, but this is the one we found. And it seems to be working quite well over a bunch of different species.
Okay, my immediate thought on that is that a bacteria strain that is commonly found in a closed, like home aquarium system, or would you have had to have sourced this out somehow? Like, is it a common strain?
I mean, that the group or the genus is pretty common on CCA. That particular strain we isolated from CCA from Guam, and our colleagues now looked at CCA from Zanzibar, from other places around the world, and they could also find quite a number of very closely related strains, not the exact identical one, and now they're in the process of evaluating these strains are producing the same compound in the same.
But we haven't actually looked for it in our aquarium.
Interesting.
Well, but it might be. There is a very common group that's got ultra.
Yeah. It sounds like it is a pretty critical, bacteria to have probably in our systems. I mean, I wonder past the settlement phase
I don't want to
what the
say
role
it's
is.
the only one. There might be a bunch more that produce compounds. It really doesn't have to be like, I'm actually sure, like in the nature, it's not just that one bacterium or even that one compound, but we found one that works well in the lab.
Yeah.
So ecologically, it might not be the only one, and probably is.
Probably, yeah, definitely.
But now, since we have the compound, the challenge is now how to apply it in a way that I can use it in a targeted way, meaning I use the compound and I expose the larvae to it, and ultimately after the larvae settle on a substrate where I want them to, on a substrate where I can rear them, that I can produce in large numbers, and it's easy to handle and so on. Yeah. And the CCA it's very easy, or not super easy, but it grows fairly well in aquaria, so you can use ceramic tiles or what have you, and have the CCA grow on it, and then use that as a settlement substrate. That does work. It's not super reliable always, but it works. But you also, it comes with a whole array of problems, right? The CCA is not a big fan of a coral sitting on top of it, right? So they try to get rid of it or overgrow it. Yeah. And then you have a whole slew of critters like other bacteria, ciliates, crustaceans, like tiny crustaceans that like to eat baby corals. And I always say that the freshly settled corals are best described as tiny, helpless, and tasty. Because they're just chunks of protein and fat, and they sit there, they can't do much. So even the tiny Asterina starfish, right? They're the equivalent to the crown of thorns for the big corals. One little Asterina eat a whole bunch of baby corals in one night.
We've seen it.
Yeah, geez. Okay, so that makes me think, Jamie he'd been co-culturing these urchins to a very specific size to basically make the settlement surfaces more, accommodating to these larvae. So were you guys doing that as well, or were you trying to find an alternative way?
No, we haven't tried that yet. I think the approach is really awesome, and it's at the same time you produce corals and you produce the urchins. So that's two birds with one stone. Yeah, that's fantastic. But our approach is to use more of a sterile settlement tile based on ceramics or polymers, where you are sure that there is no competitive organisms on it. You just have your tile, ceramics, 3D printed sand, whatever. So there's different avenues we try and test out. But ultimately we want to have the larvae on a clean settlement substrate, so we don't have to deal with all that competition. Yeah,
but that settlement substrate, it sounds like you guys are putting a lot of attention into the microbiome of that surface, and maybe the porosity is probably a component as well. Like, would some of these materials be put in close to the time that a spawn happened, or would they be long established in your settlement system ahead of time?
We have both. So we have natural grown biofilms as well, but also just completely clean, just like a couple of hours in the seawater before we actually expose them to the larvae.
Okay, interesting. Yeah. And I mean, which of you found you've had better results with? I mean, there's probably pros and cons.
Depends on the species.
Okay. Okay.
Yeah, always different. You think you figured it out, and then with the next species, everything is different, and you have to figure it out all over again. So it's really surprising. Even just within Acropora, you can have huge differences, actually.
Mm-hmm. Okay, interesting.
So it can be frustrating for sure.
Yeah, for sure. Well, and I mean, obviously not having to co-culture the urchins is saving you a stage, and it looks like you're having good results for settlement as it is. Like I've seen some of the pictures of all the tiles covered in, in happy looking corals starting to form.
No, we're pretty confident that we are in a really good path. We definitely have to get even better at it, but we've made a bunch of good steps the last years that we're confident that we can be able to accomplish it.
Okay, and actually, speaking of which, we kind of talked about this at the beginning before we started recording, but can you guys kind of explain what you guys have for facilities? Because there's sort of a farm facility and then you have a coral spawning lab. So maybe just give me like a little breakdown of what you guys are kind of running.
Ultimately, so right now we're still employed at the University of Oldenburg. We're both researchers there. I've been in charge of the aquarium facility that has been used for all sorts of aquaculture projects and competition between corals and sponges and all that. So that's one large recirculating system of about 6,500 liters, around 50 tanks, where we keep all kinds of scleractinian corals, but in there we don't do any climate simulation. So every day is the same, basically. We do experiments on grow out and all the other projects that are going on that we're not that deeply involved with, but have to manage the facility anyways. And then, as I mentioned, the pandemic hit and there was no traveling anymore. So we decided we need a facility, one of Jamie's systems, basically, or three, if you will. But in our institute, there was no square meter available anymore. Basically, every room is fought upon, and it's kind of like a coral reef, right? Everybody wants the space. But there was a little space in the garden left, so we had the idea to why not ask to have everything incorporated into an old shipping container that we can then put into the garden. That's what we ultimately had done. And
so this is is this in your guys' home? Is it, or is it at the university?
So this is just behind our research facility. Okay. There's a spawning container, so we just walk back and forth a bunch of times.
No, that's all right. Yeah, cool.
But so it's like the seawater. It has the same source for the seawater.
So we have one room where we mix our seawater. So we use Tropic Marin Reef. We have been using that since 2017, very successfully now. So from that mixing facility, the big aquarium facility is supplied, and then there is also a pipeline going to the container with the spawning system.
Okay, so the water is all just a full connected system as far as water chemistry. That's easy.
So this is just where we mix it. So and then the big aquarium is one two recirculating systems, and then in the spawning container we have 3 recirculating systems.
Okay, so you just get the new salt water is essentially what's plugged in, not the systems.
So we have 5 individual systems.
Okay.
Yeah. So the mixing facility is about 10,000 liters, and we always have on hold. And then we have one pipe, basically with seawater, RODI, tap water that goes to the shipping container.
Man, that must be nice. That must be nice. You're not mixing up any salt in small batches. It's just ready to go all the time.
Yeah, we use the
we couldn't do that.
20 kilo bags. Although I have to haul the 20 kilo bags up to 2.20 meters to get them into the mix. But it's good workout, so I don't complain.
Yeah. Gotta get some of that in.
Okay, so that's the research facility where we have in the container, we do the spawning. Then we have in the main building, we have all the labs we need. We have a climate room. We have all the chemistry equipment. We have a mass spectrometer. We have extraction labs and all that, and all the colleagues that work in the chemistry field. So that's all the research part. And now we're in the process of transitioning from the research into the commercial application. Meaning that we're setting up a whole new facility on a different location, also in the same city. In Wilhelmshaven, a few kilometers away, we're already renting the location, and we're setting up now the fully new facility where we have quite a bunch of systems and will produce commercially there.
So this system will be to sell corals commercially that you are spawning, like not asexual fragmented corals, like you're going to be just purely doing it from scratch corals.
Yes, sir.
Wow.
Made in Germany.
I'm envious of what Germany's going to have going on in a few years. That's super cool. So, okay, maybe this is a good time to transition back to kind of some of the spawning science stuff. You guys have had some good results with hybridizing between species, and well, it seems like a few at least.
It seems so. I mean, we're pretty sure they're hybrids between tenuous and echinata. So the parents we got from Fiji as tenuous, right? And they also look more or less like tenuous, right? But there was a paper coming out last year. I'm sure you heard that now in Australia the tenuous is called Kenti, and then there's the Bifaria and Indo, and so on. They got split up. So the actual tenuous is just in Fiji, and I think Tonga, but Fiji for sure. And in Fiji is also echinata. And even though they look very different, they're genetically very closely related. Right? So when we cross the Fiji tenuous that we got, the offspring has a huge, I would say, variability in their morphology. So some of them have really long tubular corallites, like echinata has, and some are growing more tenuous-like. So the thought that we have is that the parents are already hybrids, and by crossing the hybrids into an F2, then you get the variation. So that's often when you do hybrids, the first generation, they all look very similar. And then when you cross these again, then you get the unequal distribution of who gets which traits. So that's basically what we're thinking what is happening right now. But we're not coral geneticists, so we don't know for certain. But all indications are that it's hybrids. But we actually have a coral geneticist took samples. And so hopefully in the near future, we'll know for sure what is sitting in our tanks.
Interesting.
But it shows the potential that is in there. And also in our operation, we plan to play around with closely related species to see where is what is a species, right?
Like, I mean, because the species is going to be a certain percentage of shared DNA, right? Like there's probably, I don't know if they're 94% the same. Or who knows, right? There's a percentage where you call them.
It's very difficult. Like we are really not coral geneticists, and I don't want to become one because I know a bunch of coral geneticists, and like the corals are they're just so hard. They're just a really small group of scientists, and it's really hard to make a decision. Okay, where does a new species start and where it doesn't? Because the morphology is one thing, and then the genes. Like sometimes the morphology can look completely different. Any person in the reef would call them completely different names, but then the genes are so close,
similar.
And to then weigh these things, the morphology and the genes, how close they look. It's just it's a bit of a nightmare.
That goes to the point of how they had reclassified Acropora spathulata along with Millipora, and I think prostrata, like a couple of those species that look similar but do express fairly differently. They actually said they're all Millipora now, which is an interesting to go that direction rather than be like, actually there are 8 different species. No, all these ones are just one. So,
but as far as I'm not too deep into that, but I know that there's a lot of work being done on Acropora right now, and I think ultimately in the end it's going to be like something like 600 species or some insane number, because it turns out that there is also a lot of regional speciation and species that were thought to be having a huge range are suddenly split into really small regional populations, right? And then you have the species that just exists on one atoll or one side of one island chain. Also makes it difficult for conservation, right? If you only have the population or one species on one side of the island, you're one bleaching event away from extinction.
Yeah, that's a good point. Okay, so back to the hybridization thing. So I think you were saying that you had the tenuous and echinata that were obviously accepting each other well, but that may have been because they were kind of a hybrid in the first place. or genetically so close that, so this was something.
So the parents, they really looked alike.
Yeah. I mean, it's very much like human offspring. It's like you could have, parents with different hair color and eye color, and then the kids will get a combination of both. And I guess it kind of surprised you that some of the morphs were as different as they were.
Yeah, that is really the fun thing, I think, about this approach in general. I mean, this is like really the first time that somebody is really trying to put out large numbers of sexually produced corals. And just like with any other animal or with any plant, once you go into the sexual reproduction, you get the whole variety. You can cross individuals that look very interesting and that you want to bring out more. So yeah, I think it opens up a lot more diversity in our tanks. So you see, like with the tenuis, they look alike in color and in shape, and the offspring, they have a huge variety. They all look very different, and we also see that with other species as well, where honestly the parents look a bit boring, but then the juveniles, they all look different, and they show all the color variation they are able to produce. So it's really cool.
That's exciting, but I guess the thing is from settlement to actually starting to look like what it's going to be as an adult, like what's the timeline you're looking at? Because that's not going to be a short amount of time.
Depends on the species. So often the Millipora, they have this really neon green. They produce a lot of the green fluorescent protein in their juveniles up to like half a year, and then that starts to fade away, and then they get their more or less adult coloration. But they did change it over time for sure. And for some species, so one example is the Acropora austera. So we had a big spawning last October of two green parents. Basically, the mother, or I say the mother because there was one huge genotype. So we have probably 30 kilo of that one that spawned, and the second colony, the second genotype that spawned was one that was maybe like this size.
Really?
And released, very very small, or comparatively small. Released just 100 or more bundles, so just enough to get the sperm and then fertilize a huge number of eggs from the other colony. So I know who is the dad and who's the
cool.
So one is neon green, the other one is normal green, but the offspring is also
purple and blue.
Quite the diversity.
Really? So there's colors in the offspring that weren't expressed in either of the parents?
At the moment, because that's the first time that we are hearing them. I couldn't find basically much about their juvenile coloration. So they're now, they were spawned around 9 months. So at the moment, there's quite the variation.
Okay, but, so how large are they at 9 months?
Days or if that will change again. So it probably depends on the species. And
that's exciting though. So what size are they at this 9 month point from settlement? Okay.
So there's some videos from the interview. I had a bunch of them in the in the exhibition tank that we had there. So like that size.
And we already have some pictures of what they look like right now on Instagram as well.
Okay. I'll show them along in the video here while we discuss.
And then we aim for the tenuis. I mean, there's still potential to increase the growth of the speed of growth. For the tenuis, we look at about 1 year after spawning to have them at like 4 to 5 centimeters, which would be the size we would give them into the market. But we also don't want to rear them too fast, right? They're supposed to be healthy and have a strong skeleton. So we don't rear them at super high alkalinity. Alkalinity is usually a bit on the lower end compared to U.S. standards.
Okay. I'm going to get into some water chemistry stuff in a bit, but I'm curious if any of these, baby corals you've grown, have they gone out into any hobbyist systems, like as a test, or have you kept everything to yourself for now? So there's a plan. There's a plan to release at a certain point. You're just kind of gearing up for that. I was going to ask if some of these corals that were captive raised might show more potential to be a lot hardier in captivity. I mean, that's kind of the goal.
That's what we see and what the assumption is. They have been under aquarium conditions from second one. They've never seen a reef. They've never experienced ocean conditions, right? So they only know the the conditions of the aquarium water chemistry and the prevalent pathogens and all that.
Mm-hmm. So
compared to the imported broodstock, so we still get some of our broodstock is still imported, of course. I mean, they have to come from somewhere. The offspring is a lot less susceptible to like bacterial infections and fluctuations in water chemistry. And also the ones I took to interzoo, the big ones are 2.5 years, mm-hmm. and they're pretty decent size, and they were doing fine from the first day to the last, a nice polyp extension. Mm-hmm. So I think, with the imported animals, I probably would not have dared to try that.
Yeah. So you kind of knew you could get away with. I mean, you probably haven't lost any. Have you lost any? Like, have you been pretty? Yeah. So they've been hardy.
At the interzoo, no. But of course, in our research, we have.
But once they're like over the hurdle of like 3-4 months, they're pretty hardy. Like in the beginning, we lose a bunch when they're tiny. But once they're a couple of months old, we use very, very little actually.
Okay.
Yeah, the early bottlenecks are the big ones. Right? So the settlement is a big bottleneck, and then the initial survival, they start to calcify after a few days. So depending on species temperature, like after 2 days, they start to calcify and start to take up the zooxanthellae.
Yeah.
You also talked with Jamie about the uptake of zooxanthellae, which is a crucial step in the life of the coral. Yeah. So we also feed them early on. As soon as their gastric system is developed, after about a week, they can take up or they can take up food. Basically, that's also the way they acquire the zooxanthellae. But in the aquarium system, the density of zooxanthellae is really low with the UVC and ozone if you use it, and skimmers and particle filters and all that. So the natural, let's call it natural, density of the free-living zooxanthellae in the aquarium system is really low. And you can compensate by feeding them, but if they don't take it up by like week 3, 4, 5, then the survival goes way down. Mm-hmm. So we basically grow zooxanthellae outside of the aquarium and then add them at the time to really ramp up the numbers.
You just answered so many things that I was going to ask about the acquiring of the zooxanthellae, or I guess symbiodinasia is kind of the more proper term for it, as far as I understand. Yes. Okay. So because it's and also, like, I mean, I always thought it was one strain, but Jamie sort of clarified that it tends to be a few different strains.
Yeah, there's a whole bunch. I think there's how many, more than 100 are known in total, but
that's also like, we call it strains because we don't even want to start what is a species.
Yeah. So if you guys are culturing it separately, that kind of brings up a question, like, are you focusing on strains that you think are maybe more resilient to temperature swings and things like that, maybe a little bit hardier when it comes to bleaching events? Because that's the part of the science that I can see applying to, restoration efforts.
No, we definitely seen it. There's one study that's not published yet, but we definitely want to. Where we also raised these juvenile corals under elevated temperatures, and we found they can resist. Like, we had juvenile corals under 31 degrees Celsius for a year, which is, I don't know if you think in Fahrenheit or Celsius.
I think in both. I mean, it's funny, in Canada, we were on the metric system, but then we just, I'm 6'3", I mean, I say things in feet too. I don't
know. So that is basically, that is a temperature where the parents of these juveniles, 100% of them died within 3 weeks. And they can withstand these temperatures for a year without actually like any more mortality than the juveniles that we raised under 29 degrees Celsius.
Yeah.
So we definitely see that we really think that these juveniles can be much more hardy, especially when they pick up different symbionts. So we already know for a while now that the same species of coral can harbor different strains of algae. And depending on what algae they harbor, they can be more or less sustainable to bleaching or to higher temperatures. So we know a bunch of the strains that are more hardy, that can tolerate these higher temperatures better. Unfortunately, they usually grow slower with those with those algae strains. So, and that's actually also what we saw. So they survived just as well, but they were much smaller.
Interesting. I wonder why that is. That seems like such a specific.
Yeah. So we believe, scientists believe they just give the coral less, basically.
I see.
Give less of the energy they're producing. So, or at least that's one main.
Everything in life comes at a price, and if you want to survive at Celsius, I mean, you have to pay for it, and then they're smaller.
We know that there is a huge variety of algae that can tolerate 26, 28 degrees Celsius really well, but there's very few actually that can tolerate the higher temperatures. So for like reef restoration purposes, that's absolutely what people should look into. Once we start in that field, that would absolutely be what we would focus on. But of course, that's not what we want for the aquarium trade. We want the corals to grow faster. We want them to be healthy and happy and have the symbionts that they are just most comfortable with and most healthy with under like cooler temperatures where usually people keep them, and those strains would not be the right one.
Yeah. Okay. But and just a little question: would different strains of zooxanthellae affect coloration, or is it generally always brown? I mean, that's kind of my understanding from talking to Lou Eckes. Like he sort of said, you've got the carotenoids and the carotenoids and the chlorophyll, and when you get those things together, you basically get brown no matter what. But
some variation of
brown green,
that come from the coral.
Yeah, so I think that it might do a little bit, but the bright, beautiful colors that we see, that is the coral itself and not the symbiont. Yeah. So, but these are the fluorescent proteins. So this is basically their UV filter of the coral, the sunscreen of the coral, and there's a huge variety, even within many species. I mean, that's what we see, right? If you think of a tenuis or kenti or whatever you want to call it now, but we know they can come in so many different colors, and even within the same reef, you can have individuums with very different coloration, and that's just their own ability, that is in their genes, and that is also what we want to find when we cross them.
Yeah, definitely.
So some of the green and some that produce more of the pink, and that is really beautiful to see.
So again, I'm so curious on this hybridization thing. When I talked to Jamie, he said that Millipora was he called it the dirty girl of the reef, kind of the one that maybe is more accepting of different types of different species. So, I mean, you're probably finding, I don't know how many different species you're spawning right now, but are you finding that, some are compatible and some there's no compatibility? Like Millipora is obviously a little more accepting of different species, but what have you found there?
Yeah, with a variety of species, we have around 10 species. So there's always, or sometimes the case that you have one colony of one species spawning and none of the other conspecifics, but one other species. And then, of course, we try to hybridize them. But so far, we haven't had any luck, because usually it was a tenuis and a muricata. I mean, of course, I don't throw everything away. I just put them together and see if everything works out. But so far in that regard, we also had Nazuta and Humilis, and that didn't pan out. Tenuis, Millipora. So the accidental crosses we were able to do didn't work out so far.
Okay.
But with a new facility and more space, and also the focus on that particular questions, then we'll try to cross closely related species.
Yeah.
So that hasn't been the focus yet, but now looking at what happened with our presumed tenuis, it turned out to likely be a mix of two species. Of course, now that's on the high up on the agenda.
Yeah. And what we could also imagine is just using corals from different regions. Maybe, for example, a tenuis and a kenti, which we used to only call tenuis, right? They are probably pretty similar. We could try to put these in the same system and cross those and see what happens there. That would be scientifically very interesting, because that is like one answer of like, is it a species? Is it not? If you can cross it. Yeah. Or how well you can cross it. That is scientifically interesting, but of course also for us, and to see how the offspring looks like.
Yeah, and I mean, another thing that's just interesting is if you are just spawning different genotypes of the same species, like you were saying, you may still get offspring that don't look like the parents or just have some wild variability.
Yeah, exactly. And then there's like almost nothing known, I would say, to the heritability of coloration, right? And the color protein from Acropora, I think there's like 90 different proteins have been identified so far. There's probably many more, but as far as I know, there is no knowledge about what happens when you cross, like on a scientific basis, what happens or how is that going through the generations.
Well,
to figure out what happens across the green and the yellow and
my kind of thinking on that, what makes sense to me from just thinking about how nature works is it's creating that variability intentionally just to add diversity, so that if one color, maybe a fish is more likely to pick at that type, and maybe it'll pick at the red Mellipora but not at the green one. If it has the ability to vary what its offspring will look like, then that increases its chances of survival slightly, I guess. No,
it's a really good question. Like, why did they not just come up with one sunscreen? I mean, it's just UVA and UVB. It doesn't really change, right? Why do they need so many different compounds to protect themselves? It's really, really curious.
There's definitely more to it. Yeah, and diversity is always key to survival.
Yeah,
that's what we're always seeing. Regardless which organism. If you increase the diversity of the population, they always have a better chance of making it through tough times.
Totally. Yeah. And not just color variants, but like you say, growth variants is a different growth strategy. Like the Echinauta's got the polyps are more spaced apart, and maybe it's able to grow, out faster than the tenuous is or something like that, which, may give it some competitive advantage, but it comes at a cost in some ways too, right? It's not as strong and rigid. So, easier to break in a storm or from a fish crashing around or whatever.
Exactly.
Yeah. Okay. So, on the sort of spliced coral, chimera coral kind of thing, I'm curious if you guys have had any mixed success with stuff like that.
Yeah. You mean like early fusion?
Yeah. So, I guess, again, this is another thing. If people want to see the specific details on this, I talk about it in the episode with Jamie, but I'm curious if you guys have had results with that too.
Yeah, we also have chimeras. Also right now we have a bunch, and that is also a focus we want to look into, up to which age are they able to fuse, because it seems like in the early phases after settlement, the immune response that tells them, hey, that's not me, right? I should fight that guy. That seems to be switched off, so they can fuse and then form the chimera, and that makes them a bigger colony at an earlier time, and they can share nutrients and all that. And then, of course, and also you have if it's two color morphs, then you have two like a spliced coral with like two colorations. So that is, of course, very, very intriguing, and is also something we want to go more into, to do it in a targeted way to design, create multicolored genotypes or colonies.
Yeah, and that's probably going to happen more when you have a lot of, probably easier if it's the same species spawning at the same time, and you can isolate, some from one and some from another. But I guess it's still complicated to, know what you're putting together at that point, right? When you're collecting from a spawn that's, say, you have 10 different Mellipora spawning.
The same species, right? So I'm not aware that there is chimeras of different species. So it has to be the same species but different individuals.
Yeah. So when they grow together, I mean, we could try, but I wouldn't be very hopeful, because we already see like it sometimes works well that they grow together and grow, and like you can see one colony with like 2 or 3 different colors on different sides, but often it doesn't really work so well. So I wouldn't be too hopeful that if larvae from 2 different species settle close to each other.
So they really do have to be the same species. Yeah.
Okay.
Yeah.
And then
this one, I would think, like, we haven't tried it. Like corals, they always surprise you. You think they're so simple, but they surprise us every day, and they do things differently than we would think. So maybe.
Yeah. definitely.
And then what helps with that is when you can't settle them in a targeted way, so they actually settle where you want them to. So you have to distribute it evenly on a substrate, and then you can put them together in a targeted way and pick the different color morphs to see which one can fuse and which can't.
I mean, for you guys, there must be some fun to just, having a bunch settle and just seeing what happens too, because some of them may settle next to each other, some of them may not, but you're going to see what happens as they start to grow out. Yeah.
Sometimes there's too many settling in one spot, so they tend to, because they benefit if they create the chimera, especially in the beginning, that they're bigger. But if you have a blob of like 50 larvae that settle next to each other, and you have a chimera, or that they fuse together early on after a few days into one, they're usually not going to make it. Then if it's there seems to be a cutoff. I can't say where that is, but if it's more than like 3, 4 or something like that, then
so that's kind of when the immune system.
So we also had them sometimes settling in patches. Then you have like 100 or 200 in one spot, and
it's kind of sad to see, because it kind of looks cool in the beginning.
Yeah, you're like, holy crap, look how many settled here. And then they're just all fighting at a certain point, right?
Yeah, or if they
pretty early on, they die off, actually.
Yeah, that's hard to see. But I mean, you guys are obviously having good survival rates at this point. But it's been 10 years or so you've been kind of at this for, just about?
Yeah, more than that.
Yeah. Okay, so no joke. It's a serious endeavor, and you guys have the education behind it too. So, yeah. Okay, so I guess I want to talk about coral health a little bit, because we can all assume that if you are getting corals to the point of spawning in the first place, they are extremely healthy and have, excess resources to produce these egg and sperm bundles. So kind of take me through maybe a little bit of your feeding protocol and how you kind of approach nutrition and nutrients. And we're just talking specifically SPS or Acropora you've been spawning, right? So, yeah.
So for the basic water chemistry parameters, we don't do anything extraordinary. I would say we usually have alkalinity between 7.5, so rather on the a bit on the lower end. What surprises some people is that we have the phosphate usually a bit higher, around 0.1 milligrams. So they seem to really enjoy that. So when it goes around 0.05 or lower than that, then I get a bit nervous, because when you dip into like the really depletion, then they usually don't respond too well. So I like to have the phosphates a bit higher, nitrate not so much higher, around 1 milligram in that ballpark number. For the feeding, we use, commercial food, but we also mix our own. Basically, we take everything that lives in the ocean and run it through a blender, and then they can pick whatever they feel like, basically. So that's something we have been doing for quite a while now. So we prepare it ourselves, but we also add stuff like the Arctic calanus, copepods. Then I used to do phytoplankton filters. Sorry.
What happened?
Sorry.
At the moment, we don't have the time for doing live phytoplankton, so we use concentrates and add that into the feed.
And then otherwise, the food is just a mix of seafoods that you blend and freeze.
Smoothie.
Yeah, basically
slurry.
And then ideally, it's being fed at night, so that's their natural feeding time.
Okay.
And it comes quite handy that in the spawning systems, the days are set in a way that sunset is around noon, right? So spawning doesn't start in the evening, but early afternoon. So you get home at a reasonable time. So that also enables us to just regularly feed the corals when they're responding the best to food at night. When they're all open extended, then we can provide them with the food. And one thing also that, in my experience, that it shouldn't be overdone with the food, not too often, because if there's too much organics, or they take up too much organics, I think at some point their microbiome is getting a bit out of hand.
So the frozen food, do you feed it every day, or is it alternating days?
It's alternating.
Yeah. Okay. Because I've actually wondered recently. I do feed a very similar mix to what you guys do. I blend a bunch of seafood. I add some mysis and brine as well, and then sometimes I put calanus in there, just like you guys. But I've actually wondered about almost putting too much amino acids, or some of these fatty acids, like maybe a little bit too much, maybe causes issues. Have you kind of found something like that? I think it's kind of what you're just saying.
Yeah, I mean,
really.
I didn't do it like in a replicated, scientifically sound way, but in my experience, if you feed them too much, they tend to be more susceptible to bacterial infections that are hard to fight. I mean, they always respond really well to the food, but if I drink 5 shots, I also respond really well. But if I do that every day, it's not going to pan out in the long run.
Yeah, that's true.
Just maybe not too fitting comparison. So I like to feed them, but not like continuously, but like.
And I guess an important question is: are these systems stocked with fish as well? Like, is that part of it?
Yeah. So we do have fish, but usually the basic cleaning crew. Like the Acropora, the filefish, then a bunch of copper bands and herbivores like standard sturgeons and rabbitfish and all that. So the fish stocks are not super high. So we also supplement ammonium to provide the zooxanthellae with the ammonium that they need, and phosphate. The phosphate we add as recently the new product from Tropic Marin, the phosphate.
Phosphate, yeah.
That provides the phosphate in a particulate form, so you don't, do a blanket dosing that every organism can use, but more targeted, that the phosphate is available to the coral in the coral, basically. So that I'm quite a fan of.
Okay, yeah. So your phosphates would probably go to 0 if you didn't supplement in some way, though, I'm assuming.
Yeah.
Okay. Yeah. And I've tried that phosphate.
We just have a lot of coral compared to the fish we have.
The coral per water volume ratio is high. It's similar to my systems too. Yeah.
We have some tanks where there's barely water in.
Yeah. Okay. Yeah. So the phosphate is definitely a good product.
Dosing, like the normal dosing system, you do the classic barling system. We go through a lot of volume. I think the big system is running through about 3 liters of barling solution per day, which is quite a bit. But on the other hand, it's very easy to prepare. Right, a calcium reactor would probably maybe be more, cost efficient, or I don't know. I haven't done the calculations, but a calcium reactor is also not as easy to run.
Yeah, to dial in.
We have people working also in the aquarium that don't have the aquaculture background. So if I tell them, you take a bucket of water, you put in 10 liters, and you put X amount of that powder and stir it, and then you put it into the container, then everybody can do that. Yeah. And if you start CO2 bubbles and that stuff, and it can be a bit critical, especially if you have high consumption of alkalinity. So some systems are up to like 2 degrees per day. So if you run into problems, and you miss 1 day of dosing, then
I agree. Especially calcium reactor does have a tendency for the line to get blocked, and sometimes it can just be just a little bit and you might not notice. And I don't think there's flow sensors that detect such a low flow rate particularly accurately. So you'll just notice maybe the stream, does the stream look a little thinner or a little more dotted than usual? I agree. Obviously pros and cons to every system.
Barling system, you can't really go wrong, in my opinion.
And then you guys are adding the A and K trace elements as well.
Yeah, A and K trace elements. If there is a shortage of some element, we also do that in a targeted way. We add extra fluorine or copper, nickel, zinc, what have you. Then for monitoring is also a big issue, of course. We have the GHL sensor system in our big system, right, with the pH, redox, salinity, etc., that I can see remotely. So I'm pretty happy with that. And then in the spawning systems, we have the Apex systems. that give you live data, basically. For the dosing, we have the alkatronic, dosetronic, but we don't let the alkatronic do the
dosing,
decisions of the dosing. So that's more for monitoring, and then the dosing I dial in by hand, because I mean, it's all, the technology is all nice, but if there is a problem and the probe has an issue.
I wouldn't want to trust the machine in that case. Definitely. Yeah. So are you guys doing fairly regular ICP tests on some of the systems? Yeah. Who are you
usually
doing that with?
once a week, actually.
Okay.
Or every 2 weeks at the latest. Yeah.
That must be nice. Which company are you doing it through?
So currently we send to Fauna, but Tropic Marin is bringing out the ICP. So.
Oh, interesting. Okay.
Decision is clear. It just came out at Interest was presented that they're having their own ICP line, and also the element dosing system. That is just coming onto the market.
Yeah, I'm surprised they're a little late to the party on that, actually. It seems like. But
not that easy to set up, and there's already a bunch on the market, so you want to be good at it.
Yes, definitely. Do you know if it's going to be MS or OES? Do you know what the system is?
OES, but there is a new dosing system, or for corrective and maintenance dosing, where you don't have, they have the single elements, but also combined elements, like the elements that are set up in groups, so-called blocks. So like the halogens are one block. So we know usually they're used up in a certain ratio. So if I know iodine is being used more or low, then the other halogens are probably equally low. So I can dose all of them in one go. And the nice thing about that is that the ICP, the OES, is not as accurate in the low range, and there's some overlap of signals. So sometimes it's hard to figure out if the number you get is actually the number of what is in the water. So there's the block elements. There's some indicator element that is good to read, that can be measured accurately, and based on that reading, the other ones are recommended.
Yeah,
basically.
Okay, that sounds like a cool way to do it.
Trace method can add all of that.
In
one go.
Yeah.
So that reduces 40 bottles to just 5.
Yeah.
Which makes complexifies the whole thing.
Yeah, because as great as the moonshine method has been for some people, it's definitely like a daily, very involved method. So I do like the sound of that. But at the end of the day, whatever works, right? Whatever works for your lifestyle and whatever works for your tank is
exactly.
So it's nice to have different options out there.
Yeah, and we have to monitor quite closely as well, because we have so much consumption, and then also when we simulate the temperature and light curve throughout the year, the consumption varies a lot by the seasons. So there's just a huge variation all the time in our year simulated system. So there you always have to have a close eye on, like, if the consumption changes.
It's not always the ideal conditions for the corals, to be honest. So in our Saudi system, so if we would use the original data, it would go up to 32 Celsius in summer. So we cap it at around 30, but still you can clearly see that the alkalinity consumption and all that goes quite down in these stressful times. Also in the Australian system, it goes up to 29. Winter time right now is at 24, so that's quite the stretch.
But there must be a temperature where growth is actually, where it's a little on the higher side and growth is at its fastest, right? Because I would imagine you're essentially, it's like faster metabolism essentially, but at a certain point you start to see a drop off where it's actually the coral is slightly stressed, so the growth slows down. So you say is that around 29 or 30 Celsius kind of thing?
That really depends on the coral.
Depends on the species too,
Yeah.
Exactly. Depends on the coral species, where they come from, what algae they harbor. So you can't really say this is the perfect temperature for all scleractinian corals or even all acroporates. There is no one answer for that.
Okay. Yeah, interesting. Okay, you mentioned that you were dosing ammonia, so I wanted to get into that because that's a little becoming more popular in the North America now too. How long have you been dosing ammonia, and kind of why switching to that from nitrate dosing?
It's been a couple of years, at least, I would say. Yeah, like three, maybe more years.
Okay.
So I use ammonium, NH4Cl. Basically, and that's the preferred form of nitrogen for the zooxanthellae. And they can also utilize the nitrate, but that is like energetically more expensive to utilize. So the NH4 is what they like. So we dose 2 times a day. There was to see if I can find. There was some publication that looked into ammonium concentrations in the reefs, and there's usually 2 peaks at a day. In the morning when all the fish get active and feed, and then they excrete, right? Then you have 1 peak, and then the same in the later in the day. So the concentration in nature is not always the same, but there's also peaks where they have more available.
I was just going to say, okay, that's interesting. So there's 2 ammonia spikes in the day. It's kind of like almost like, if people go take a number 2, twice a day or once a day, there's some kind of consistency to it, right? But so maybe that there's some corresponding peak in for some reason, right? Although I imagine fish are just kind of constantly just going
maybe
whenever
when they
want.
wake up.
Yeah, who knows, right? But either way, there's a peak. So okay, so are you using ammonia bicarbonate? Is that your go-to, or some other form?
Ammonium chloride.
Ammonium chloride.
NH4Cl.
Okay, cool. Yeah, I think you cut out a little bit when you were saying that. So
Ammonium chloride.
Okay.
Just dissolve it in RODI, and that's
yeah, cool
solution that I use.
Okay, so yeah, obviously better bioavailability, and also the coral can utilize it a lot easier than nitrates. And then whatever doesn't get used of this ammonia would, in a good established system, with a lot of, biology happening in it, like maybe larger biomass, that extra ammonia is going to get converted into nitrates pretty quickly, right?
Exactly. Yeah. So that's also the way you can track your if your dosing is right. So if your nitrates are rising up, and then one thing you could do is reduce the amount of ammonium you put in, because that's the end product is in nitrate. Okay. So yeah, exactly. That's what would happen.
And I'm curious, do you, from the science kind of side of this question I've been kind of bouncing around for a while, is the ammonia being consumed by the zooxanthellae, or is it being consumed by the coral polyp or the coral itself? Like, how's it getting utilized?
So the ammonium is utilized predominantly by the zooxanthellae.
Okay. So algae in the system is potentially competing in a way too. Like other nuisance algae could potentially be competing for ammonium at the same time.
Potentially.
Yeah. Just one thing I would say, because I switched to dosing ammonia maybe 3 months ago from dosing just straight nitrate. And I actually noticed less problem algae in the tank, like a reduction of nuisance algae. So to me, it seemed like the zooxanthellae was almost better at utilizing the ammonia than the problem algae was. But I mean, it could have been other factors at the same time. But I just noticed my racks and everything looked quite a bit cleaner in a fairly short amount of time.
Nice.
Yeah.
Cool. What kind of ammonium are you using?
Ammonium bicarbonate. Yeah.
Okay.
And I just split it into 3 doses. So there's, just to not put too much at a time, obviously, because there's a danger to the fish. So it's 3 or 4 doses a day, pretty low concentration. And and I mean, I just test my nitrates regularly, and they sit in a kind of an okay range. But you guys were saying your nitrates, you keep them fairly low, like around 1. Yeah.
I mean, sometimes they also get higher, but ideally around 1, 2, 3.
Yeah. Because I guess really at the end of the day, what you're just seeing is the excess that's left over after, the corals have taken what they need, right? So, in a way, but so because based on what, if that's 1 and then your phosphates are 0.1, you're kind of more like at 100-to-1 kind of ratio, or, well, no, that would be, that would be
10-to-1.
That'd be 10-to-1. Yeah. Whereas a lot of people talk about 100-to-1 as being the ratio, but it sounds like the 10-to-1 ratio is okay too. There's sort of some acceptance in that range.
Yeah, I think Above 1, I think there's a limit to the nitrate that can be utilized by the zooxanthellae. So if it's above 1 milligram, it probably doesn't make much of a difference to them, except you have the negative aspects of higher nitrates. So
if they have what they need,
I think that the driver or the driver of the whole thing is the phosphate availability.
Yeah. What do you think in terms of like coral polyps taking in, what they need? Like what percentage would you say is, particulate food versus bacteria, which is energy it gets from zooxanthellae? Like, is there kind of a rough guesstimate of what, a coral really needs?
Yeah, it really depends on the species. So some definitely need more food. Others only need very little. So, and I think they can also do with a bunch of variety, just like we can eat a bunch of carbs, or we can eat a bunch of protein, and there's not just that one answer that is right. There is a variety is probably fine, but it definitely depends on the species.
Yeah. Okay. But it sounds like you guys are kind of doing a little of everything, so it's kind of covering the base as well. But do you? Yeah,
so we don't give bacteria food at the moment at all. But when we do the smoothie, there is a lot of different sizes, or like particle sizes. There is a whole bunch in there. So.
Yeah, because there's a lot of discussion going on right now, especially in North America, about kind of infusing some of the foods that go to the coral with different strains of bacteria, so that it's kind of like a way of the polyp taking it in. But I'm curious what you think.
Sure, yeah. I mean, there's a lot of research being done on that, on the probiotics, and also how they might help corals to become more resilient to stressors. I mean, people in Saudi Arabia had good results with providing the corals with the probiotic bacteria, and that made them more resilient. So I don't know the details of that, but there's been studies published. So in my view, there's definitely something to it if you have the right type of bacteria. I mean, we benefit from probiotic bacteria as well. Yeah. So the corals certainly would as well. It might also be a way to combat pathogenic bacteria. Right, to bring the microbiome back into shape, or to outcompete the pathogenic bacteria in a way.
We definitely, like, the science field is just starting to realize, we know that the bacteria are really important. I mean, we talk with the coral, we talk about a holobiont. We don't just talk about the coral, like the animal, but for a while that the algae are just as important. But we also see the bacteria as part of this holobiont, and we're just starting to figure out what all the bacteria in the different compartments of the coral are doing, what their roles are. Yeah, we just know very little so far. Yeah. But it's definitely a very interesting field that I follow a bit as well, and I'm sure like within the next 5-10 years we will find out a bunch more, and we might conclude that we need more of this in our tank.
Well, I wonder, I mean, it might be an avenue that you guys go down in terms of some product development eventually, based on where this farm goes, and developing a food goes along with your corals you sell. Or
it's definitely on the agenda to also work in that area.
Just wanted to see if you had any observations in terms of lighting and schedules and peak periods and things like that. So can you tell me like what kind of lighting you're running, and a little bit about the photo period?
Sure. So in our main facility, where basically every day is the same, we use predominantly the radions, mostly the generation 4, which I think are my favorite lamps to be honest. The moment, but also the newer generations we use as well. So the schedules we put together ourselves. So usually the photo period is about 12 hours, and depending on the corals and on the experiments and what we're doing, the intensities can be as low as 80 and can be as high as 500. So there's quite a bit of variation.
You're talking par, up to 500 par.
Yeah.
Okay.
Yeah, exactly. Yeah.
And so peak period, how long would your sort of peak light period last for, and how fast would it get to that as far as ramp up time?
For the highest intensity, And that's where we had the ones that I took to interview that goes after 3 hours. It's 11. It's about 2 hours peak period where they have close to 500, and then it goes slowly, go down in a linear fashion.
And then spectrum probably gets more blue towards the end, obviously, too.
Yeah, but it doesn't change too much to the blue. I don't do an actual blue phase at the end.
Yeah.
So
okay.
It just it ramps down, basically.
And how fast would you get to peak period? How much ramp up time?
Within about close to 3.5 hours.
Okay. Interesting.
4 hours. Yeah.
So a bit of a slower ramp.
Yeah. And in the spawning systems, we use the Reef Bright light bar, LED bars. Mm-hmm. It's a British manufacturer. It was built in England, so it's kind of logical to use a local lighting system. So these are nice, and we have how many par on the top is about 300, depending. Also, they're not all the same distance to the water, but I say around 300.
Yeah.
And the peak period is, so it ramps up. It's 4 bars over each tank where the mother colonies are in, and then they're sequentially switched on. So it's not like a linear ramping up. Like one switch is on, and then the next, and then the next, and so on. So that is after. I think we have it at the moment also after 4 hours that we have to peak. Mm-hmm. And then that lasts for another 4, and then it ramps down again.
I'm surprised that you're using bars that just come on and off on the spawn. This is on the spawning system, because I would think I was thinking you would want more like a really gradual curve to the ramp up, but I guess
it's more about the photo period, and then to stick with when it should be dark, it should be dark, and when there should be light, there should be light. But whether or not it's linear, maybe it might help to optimize things, but so far for our purposes, it has worked the way it has been designed. So it's run by the Apex computers, and then they just sequentially switch on and off.
Okay. Yeah, and I assume these bars are like a blend of spectrums, like they're 50-50 or something like that. Yeah. Okay.
Yeah.
Cool. Okay, well, that's interesting.
Aqua Perfect is a German manufacturer. They also do the LED light bars. That's the lights we had over our tank at Interzoo. Mm-hmm. Very, I would say, simple sounds a bit derogatory, but they're without an app, so I don't need the 48 app on my phone.
Yeah,
it's just and the spectrum is good. I like those quite a lot, so we've been using those more and more.
Cool. Okay. Yeah, I kind of like that. I like products that are basic like that. I like things that just can be controlled with an outlet sometimes.
Yeah, exactly. But the nice things about the Radions is that you have all these channels that you can individually program, and that is there's also something we want to explore more in terms of how the coloration of the juvenile corals is expressing under different lighting systems.
Yeah, and
light settings. For that, the Radions are fabulous. No doubt about that.
And one thing I've kind of thought about before is that like a coral that is at its healthiest to spawn isn't always going to correlate with the best coloration at the same time, right? Like, would you agree that maybe sometimes a coral that's like the most healthy, let's call it fat, maybe, like ready to just produce whatever it needs to, might actually be a little bit darker or not quite as appealing visually initially?
Yeah, sure. So also the aim is to have the mother colonies to be as happy and healthy and strong as possible. So also the feeding is very necessary to allow them to produce all the eggs and sperm, because if you think about the amount of fatty acids and proteins that are being released during the spawning, sometimes it's really insane. Or the Austera, which we have like 30 kilo sitting in the tank when that one spawned, the amount of stuff that came out, it's mind blowing. So you definitely don't want to miss it, because that would have bombed the system, no doubt about that. So the amount of energy they put into their offspring, it's mind blowing.
Yeah, and like you say, maybe they can't produce those egg and sperm bundles without. Maybe that's where those particulate
if you have
foods
like these
really come in.
If you have these half-staffed corals where you just want to see the have very few symbionts and a lot of the neon colors, those corals would not give a good result in spawning. Definitely not.
So and the idea also, our approach is like the mother colonies, they don't have to be pretty, right? They just have to provide genetic variation, and the offspring is going to show their potential, basically. And in the offspring, then you have the whole variety from just dull coloration all the way to the really popping ones. But I don't care if the mother colony is a bit more brown or not.
Still give the genetics, right? Even if the beautiful intense colors are covered by the brown algae, they still have it in their genes, so they'll still give it to their offspring. So that really does not matter what the mother colony looks like at the time of spawning. It just matters what the genes can do, basically.
Yeah, and I guess once you guys have successful spawns that you're raising to sell, you're going to have a system that's more catering towards pulling out the best coloration on those offspring.
Exactly. And then also ultimately we want to continue breeding with our own broodstock, basically, so we're not relying on always getting in new colonies, but to have our own lines, basically.
And do you plan on, especially because originally you're going to want like a lot of broodstock to kind of pull from, are you kind of focused on any specific regions, or like you said you had some corals from Fiji, but are you kind of trying to get stuff from all over wherever you can, kind of thing, like essentially having a bank of corals?
That would be awesome, of course, but there is limitations in terms of space and also the logistics to get corals. So currently we have broodstock from Fiji, from Australia, and from Saudi Arabia. Also because they spawn at different times of the year. Saudi Arabia is spring, early summer, and then Fiji in autumn and or fall, and Australia in winter. So in the new facilities, we also want to look into Indonesian corals, also because of the availability. And Australia, of course, Fiji. And then there's not that much more where you could source them from.
Well, I guess your other way is sourcing them from fragmentation in the hobby too, because obviously Germany's got a lot of great hobbyists. And
that is also something that works well. If you get big colonies that are happy and stable, you introduce them to a climate simulated system. It will take a bit of time. It might be a year, it might be 1.5 years, but once it's clocked into the seasonal schedule, it will spawn at the time when that species spawns. That's also what happened to our Acropora austera. That is in our Australian system, but we got it from a museum. The museum in Karlsruhe. So originally it probably is also from Indonesia, but it clocked into the Australian cycle, and after 1.5 years it spawned.
Interesting.
That's definitely in like midterm we definitely want to completely get away from importing new corals. That is definitely our goal. So we want to totally get away.
To keep it as minimal.
Definitely as minimal as possible. Right now we already very little, because usually very few of the mother colonies die off, so we can use them again and again, of course. So, but every now and then, of course, we need a bit of a new gene pool in there. But on the long run, we don't want to continue that. And
so you might commit to a date where you're like, okay, no more imported corals. We're doing this 100% off the grid, basically. It's Cool. Well, I mean, that's really exciting for kind of the future of the hobby, and you guys are definitely pioneering a lot of that. So that's super cool. Yeah, I can't wait to see what you guys do in the next, 5-10 years. It sounds like you're, pretty committed to it. So it should be really cool.
Yeah.
Yeah.
So there is no turning back now.
There's no turning back.
And then hopefully we'll get a way to ship some of these, if we can get some of these fully aquacultured corals specified differently, maybe the CITES and the importation into different countries will be made a little more easy too.
Country of origin will be Germany then.
Yeah.
Because they are... Yeah.
Those will be the first German corals.
Yeah, and last time I checked, there was no tropical reefs in Germany, so yeah. Cool. Was there anything else you guys wanted to mention? I feel like we covered a lot here, actually.
I think we got...
No. Maybe just if anybody is interested to have a look at our facility, Toby from Sea Friendly Reef visited us a couple of times and already did a bunch of videos in our facility at the university. It is on his channel, Sea Friendly Reef, and also on the Tropic Marin channel. Yeah, so
cool. I'll put links to those. I did see the Sea Friendly Reef tour, and it was pretty awesome. So I'll put that in the description notes.
Yeah, there is a couple of English videos also on the Tropic Marin channel about the facility. I'm pretty sure. Yeah,
that's the one I watched, obviously. So yeah.
Yeah, and then maybe if you link our Instagram. So we just started our Instagram, so people can follow us and see how we built our new facility and what corals we're growing, and how everything is developing.
Totally. Yeah, you guys deserve more followers, so we'll push that, and yeah. Awesome. Okay, well, thanks so much for making time, guys. I appreciate talking to you, and maybe we'll do a follow-up at some point and see what you're up to in a couple of years or whatever. I'm going to keep doing this as long as I'm enjoying it too. So,
Awesome.
Okay.
Yeah, thanks for your interest. Thanks for having us.
Thank you so much for having us on.
Yeah. Okay, I appreciate it, guys. We'll keep in touch. Okay, have a good night.
Likewise.
Okay, bye.
Take care. Bye-bye.
Bye.
Thanks for joining me on this episode of Beyond the Reef with Sam and Mareen. If you want to dig a little deeper into the research they're doing for the ICBM program at the Oldenburg University, I will leave links to the website and some of the other videos and social media we talked about associated with them in the show description below. Just another shout out to our sponsor, Fauna Marine. Definitely check out some of their new exciting products that they brought to the market this year. If you have any suggestions for future guests, want to ask me a question, or just reach out in general, you can email me at beyondthereefpod@gmail.com. And if you're looking for high-quality aquacultured corals in Canada, please go to my website at fraggarage.ca. Hope to hear from you soon.